All right, welcome back. So now this is part two, so we'll finish the prokaryotes. I mean, the prokaryotes is what makes it different from the eukaryotes. I'm pretty sure you guys know the eukaryotes. So let's go through the rest of this. Okay. Okay. So the bacteria, oops. The bacteria do not have a nucleus, right? I'm sure you guys know that, okay? But what you guys don't know is that they have a region called a nucleoid. And that nucleoid, okay, is where the bacterial genetic information is actually stored. It's not just like floating around everywhere. It's actually stored, it's wrapped, okay? Okay. The plasmids. Okay, so plasmids are little circular loops of DNA that are in most bacteria, I think, and they carry different genes that sometimes get added to the bacterial chromosome. Sometimes they just taken out of bacterial chromosome. Sometimes multiple copies get in and out. But there's transfer between the bacterial chromosome and the plasmid. Okay, there's also transcription occurring on the plasmid, so that stuff is actually being made. Okay, they carry generally non-crucial genes to survival of bacteria in a normal environment, but it's incredibly important to us. And the reason why it's incredibly important to us is they can carry genes that specify antibiotic resistance to specific antibiotics. And those plasmids, those little circular loops of DNA, can go from one bacteria to another bacteria, and they can work. And it doesn't matter if they're the same species, different species, etc., etc. They get transferred from one bacteria to another. Okay, and that's what the sex pili does. It transfers plasmids. The other thing is there's plasmids that code for production of toxins. So if they have it, then they can produce certain toxins that are toxic to humans. Okay. The third thing, the beneficial thing, which they don't mention here, is as scientists, we play with plasmids a lot, and we can use plasmids to do a lot of molecular biology, and we do. Okay, like we put insulin genes on a plasmid and put it in a bacteria for them to make it. Okay. All right, ribosomes. Okay, are the sites of protein synthesis. Okay, they are made of protein and ribosomal RNA, and the bacterial ribosome is 7DS. Okay, don't worry about the S. What the S stands for is sedimentary coefficient, and it's made out two subunits together, 50S plus 30S. I know 50 plus 30 equals 70 doesn't make sense, but it's how fast they move through a gradient. It's not weight, okay, or math really. Okay. Okay. So small subunit, large subunit. Okay. And then this is the complete 70S. Okay. All right. Inclusions. So inclusions are little blobs of stuff, and it depends on what bacteria you have. They have different inclusions. Like for example, polysaccharide granules provide sugar. Okay, it's not like collagen or glycogen, but it's a bacterial storage form of sugar, which I can't remember off the top of my head. Okay, carboxysomes have this enzyme called Rubisco. Okay, that's for CO2 fixation. Okay, magnetosomes are actually really cool. They have they store iron oxide. Iron oxide actually functions as a magnet. So these guys actually respond to magnetic fields and stuff. Okay, see the magnetosomes. Okay, another important concept. You definitely have to know what this is. Okay, a endospore. Okay, endospores do not happen all the time in bacteria. Okay, but so when things are going good, bacteria do not waste their time making endospores. Making an endospore actually kills the bacteria that is making the endospore. Okay, so think of it that way. So the only reason why you want to do it if you're dying already. Okay, so when nutrients are depleted, when you run out of food, what you need to do is protect yourself, and then wait for things to get better. Okay, so the process of making it is called sporulation. What actually happens is the bacteria makes a copy of its own DNA, and it shoves the DNA inside this thing called the endospore, and it wraps up its DNA with a whole bunch of layers of wax, lipids. Okay, that makes the endospore waterproof and desiccation proof. So now I can't do very much inside the endospore except stay there, but I can stay there for literally forever. By literally forever, I mean like millions of years forever. Okay, so longer than humans have been in existence forever. Okay, and then when things are right again, they can come out. Okay, so here's the endospore formation. Okay, so you wrap it, and after you wrap it, you kind of kill yourself and you free the endospore. That's why I say the bacteria itself actually dies. Okay, when things are good, the endospore will germinate. Okay, so basically all the endospore is waiting for is for things to get good, and usually that's a sign. A sign of things getting good is things getting wet. Because that wet thing means things are growing and stuff. Okay. Okay, eukaryotic cells. Okay. Okay, flagella and cilia. Other than the fact that flagella and cilia are slightly different things in eukaryotes, there's not much difference. Okay, same thing. Flagella, long projections, few in number. In humans, the only place where you find it is sperm. Cilia, short projections, much more numerous. They can move using cilia, right? Humans generate current using cilia. In single cell prokaryotes and stuff, they use cilia to move around. Okay, so you can see that here. They're using that thing to move around. Okay, don't worry about this tubulins. Okay, the cell wall and glycocalyx. Okay, cell walls are found in plants, algae, and fungi. Okay, and it's made of carbohydrates. Okay, so not peptidoglycan, carbohydrates, cellulose. Okay, chitin, glucan, and mannane, and yeast and stuff. These are different, and they're all made out of sugars and stuff like that, right? But the important detail that's here, that they're not telling you is, well, being made of different stuff, even though they're all carbohydrates and stuff, allows you to attack each one separately. Okay. 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 So the same thing. The difference on the from prokaryotes is that we have carbohydrates on our surface of the cell that allows us to, one, for attachment, but more importantly for cell-cell recognition. Okay. Okay. The endocytosis is the same. Endocytosis doesn't occur in bacteria because bacteria have a cell wall that is rigid, right? So it can't fold like it. Bacterial cell walls and stuff can't fold. We can, so we have this process of endocytosis. We can actually wrap ourselves around the bacteria and bring it in. Okay. Phago, eating, pino, sipping. Phago, you actually physically wrap around something and bring it in. Pino is just little sips, okay, to bring in a little bit of fluid around the cell to see if there's anything worth getting. Okay. 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 Cytoplasm is the same stuff. Ribosomes are different. Okay. Ribosomes in bacteria are 70S. Ribosomes in eukaryotes are 80S, except in chloroplasts and mitochondria. Chloroplasts and mitochondria have 70S ribosomes. The reason why they have 70S ribosomes, just like bacteria, is because they are bacteria. Okay, they come from bacteria. Okay. All right. These next couple of slides, I'm gonna let you guys read on your own because honestly, you guys can understand this on your own through the PowerPoint. And you guys all had this in anatomy. It's basically the exact same thing. Okay. Okay. I'm gonna cover chloroplasts a little bit. Chloroplasts, okay, are kind of like mitochondria. They're also bacteria that got trapped. But chloroplasts is something different than mitochondria. They absorb sunlight and they use sunlight to make sugar. Okay, but it's the same kind of thing. You have these membranes that repeat over and over, just like the mitochondrial inner membrane does. And because of these membranes, you can make a bunch of stuff. And the chloroplasts, they're green, so they absorb light, except green light. They absorb all the other wavelengths of light, and they can actually use it to make ATP and glucose. Okay. Okay. So one of the big things I'm will definitely ask you on this is the evolution of eukaryotes. Where do eukaryotes come from? Okay. Okay. The evolution of eukaryotes. Life arose as simple organisms 3.5 to 4.4 billion years ago. The first eukaryotes evolved over 2.5 billion years ago. Okay, so here's the endosymbiotic theory. The early cells, okay, split into bacteria and archaea, but some of them got bigger and became eukaryote. So they started folding in their membranes a little bit, and that folding in of the membrane allowed them to form more surface area, and they started to form internal membranes. The internal membranes allow them to grow much bigger. Okay, much bigger allows you to wrap around your nucleus, and now I can get even bigger. Okay, that's what the endoplasmic reticulum is. The endoplasmic reticulum in reality is an internal membrane, and so that allows you to separate things and get bigger. And now I can punk smaller bacterial cells. Okay, So there is the quid pro quo of it's like the world's oldest mafia scheme. Okay, look, you're a tiny little mitochondria. I have a big huge cell. I can either eat you or you can provide me with a bunch of ATP and I can let you live inside me. And so that scheme, okay, allowed mitochondria to stay. So they pay us an ATP to stay inside our cells. And so they become so ingrained inside our cells that we can't live without them already. Okay, and this is the same for a group of chloroplasts. Chloroplasts become trapped as well. Okay, again, the same Ponzi scheme. I keep you alive, you pay me in sugar this time. Okay, so this is the model of eukaryote, the origin of eukaryotes. Okay. All right. So here, congratulations for making the end of this chapter. Okay, I'm gonna make a comment. Okay. So based on theory, endosymbiosis. That was what I just explained. Okay, we are actually from Asgard. For those of you guys who watch Marvel, this is probably funny, but there is a reality to this. That will show up as the extra credit on your exam. So if you can figure out why I said this the way I said it, write that on the exam, okay? And you will get extra credit for it. All right, good luck.